Yeah, first of all, I would like to thank the organizers for inviting me to this very fascinating meeting. It's my second time visiting the Middle East, so it's particularly exciting for me to be here. And so, as already announced, I will talk to you about heart development and the role of genetics and biomechanics, and hopefully at the end of this talk, you may feel that it may have some connection to also to cardiac disease, which I think is evident, and hopefully you will agree in the end. So, I mean, when you deal with hearts, it's usually the kind of hearts that you see here with a quite complex valvular apparatus. But I mean, if as a developmental biologist, we start with a heart that looks completely different, and that is seen here. So this is a mouse embryo at embryonic day 7.75, equally about 20 days of human development. And the red area that you see here is the cardiac mesoderm. So this is the location of the precursors that make up the myocardium. At this stage, it's just a piece of tissue, just a layer of tissue on either side of the embryonic midline. And they still have to fuse in the midline to form a tubular heart. So this is depicted here. So the next stage is this structure here, which is a tubular heart, which then at this stage starts with the first heartbeat, which is initiated first here in this position, and then subsequently the cardiac pacemakers will be generated. What is depicted here is a recent reformation of the model, because it has been realized that the heart is not formed in a single step, but by multiple steps. So what you have are two heart fields on either side, which are separated here in a blue and in a red lineage. The reason for that is that initially the myocardium is only, that you see, is only formed by this blue lineage. And this is the future left ventricle of the heart. And the red lineage, what you see here, which is initially in the midline, and then after fusion of the two heart fields will be positioned on the dorsal side of the embryo, will be a resource of additional cells, which will be added both at the anterior as well as at the posterior side of the myocardium. And with this population of cells, the initial embryonic heart will grow in size. And so these two populations are called the first and the second heart field, for those of you that look into the literature. To make this a little bit more clear, it's shown here. So the first heart field, the blue lineage, will make up the myocardium. The second lineage is sitting here at the dorsal side of the formed myocardium. It will send then cells here to grow this heart into size. And so most of the cells are migrating in this direction, and some cells also migrating in this direction, adding then to further growth of the myocardium. Another way to visualize that is here with a scanning electron microscopical series of scanning electron microscopy pictures from a chick heart, where the same process happens as well. So initially you have this small population of cells. This is a blue population that I've shown you initially, and then cells are added to extend this in length. And this is further growing, and so this second population is there to generate more myocardium. But it's also important to realize that the blue initial heart lineage forms only the future left ventricle, and all the other myocardium, so the right ventricle, which is this part, as well as the future outflow tract, the green part, are all derivatives of the second heart lineage. So it's a rather complex way of generating the myocardium. What is depicted here is we are now in this stage. We have a linear heart tube which then loops towards the right side, and if we section that, this is shown here, we can see that the histology of the embryonic heart is rather simple. It consists of a myocardium, here shown in red, and an endocardium, which is similar to endothelium of a blood vessel, which is separated to an extensive extracellular matrix, a so-called cardiac jelly. So it's a rather simple structure in this moment. And this has to be remodeled then further. What is depicted here is now, again, this embryonic heart. So the myocardial layer is out here, and the endocardial layer is here. You can see this free room, which is normally filled in with extracellular matrix. And even at this stage, the heart starts to beat, and it's able to propagate the blood in an anterior way. So, I mean, despite that the heart looks very simple and has no valves at this moment, it's still able to directionally pump blood. It achieves that by a trick, by generating a contraction close to the posterior pole of the myocardium. And this is a part of an elastic, so the entire myocardium itself is rather elastic, while the vasculature on either side is rather rigid. And what is caused then is by this contraction, a wave is generated in both directions, but because the contraction has occurred at the posterior side, there will be a reflection occurring earlier on that side than on that side, and thereby a wave is generated which is directional. So despite having no valve, the embryonic heart is already able to generate a directional pumping. Well, but this is of course not sufficient for a growing embryo, so there's a stage at which the whole thing has to be more perfected. And that is depicted here. So this is the looping myocardium. In the embryo, it's about 10.5. So at a week, about in the human embryo. And what you can see here is that the heart has now looped, and it's starting to generate the chambers. So this is a future left ventricle and the future right ventricle. And over in this area is a future atria. If we cut that open, we can see here that internally there's already the growing septum, and here the yellow pieces are is the tissue that is most interesting for us today, because these are the so-called cardiac cushions. This is extensive extracellular matrix, which acts now as a temporary solution to generating a valve-like apparatus. So those cushions are sealing the atria and are sealing the ventricular chambers to allow then a directional blood flow to occur. So those cardiac cushions, which as I say is the next best solution after the initial cardiac jelly, those cushions are positioned, one in the AV canal, so which is separating the atrium and the ventricle, and there's another pieces of cushion in the outflow tract, which you can see here. Both areas are demarcated by certain signals, which are the BMP signals, the bone morphogenetic proteins, which are expressed in this area, shown here, and they are inducing a transcription factor called TBX2. And TBX2 is both expressed here in the AV canal as well in the outflow tract region, and this is demarcating the area where the cushions will develop. So we have a little bit of genetic factors that are involved in setting up these areas of cushion development. Inside the heart itself, there's also a signaling molecule called vascular endothelial growth factor, VEGF, that causes the endocardium in this area to proliferate. And there's also remodeling going on here in the cardiac cushion itself. There's particular accumulation of certain extracellular matrix molecules like vertican and hyaluronic acid that accumulate in this area to generate swelling and also setting the stage for the next step. So the next step is now that those cushions will be cellularized. So cells are migrating from the endocardium towards the myocardium. So they're migrating in and populating this initially cell-free room and generating then the true immature cushions. This process is called the epithelial mesenchymal transition, or EMT, a process that happens in many different parts of the embryo, but it's quite prominent in the valve area. It happens both in the AV canal as well as here in the outflow tract. So in both cases, an EMT process occurs. And we know quite extensively the molecules that are involved in this regulation. And the main reason that we know so much about it is that we have a biological assay to analyze this. So what you do is you excise the area where this process occurs, the EMT, and then you place this excised myocardium together with the endocardium on a piece of collagen, on a collagen gel. And then the cells will spread out, and ultimately they will do the same as they would do in the true valve region, because they will then migrate into the collagen gel as if they would migrate into the cushion region. So this is also shown here in a microscopy picture. And then, of course, once you have this assay set up, you can then play around with it. For example, you can ask the question, is BMP important for this process? And so this is done here. You can treat this with a certain molecule that interferes with BMP signaling, and then this migration doesn't occur anymore. And so this kind of assay was very successfully used to work out the molecules that might be involved in this EMT reaction. And by now we know that there is a huge number of signaling molecules that play a role in this process, named here. So TGF-beta has been identified, BMP, which we have mentioned already, is involved. Very important, and we will go a little bit more in detail, is the Notch pathway in this context. Wnt and beta-catenin, VEGF, all these are involved in inducing the epithelial mesenchymal transition. So endocardial cells will leave the endocardium, migrate into the cushion, and then ultimately this is a fully mature conal cushion which is fully cellularized at the end of this process. So I mentioned that the Notch pathway is a very important process that plays over and over a very important role in valve development. And it's by now a quite complicated pathway, but nonetheless I thought to at least schematically introduce it to you. The main thing about the Notch pathway is that it's a pathway that involves cell-cell interaction. So only when cells are meeting each other, then this pathway will be activated. The reason for that is that both the ligand and the receptor are both integrated into the membrane. So only when the two cells are directly in cell-cell contact, this interaction occurs. And what happens is when the ligand binds to the receptor, then a proteolytic process occurs, and a domain is cleaved, which is the Notch intercellular domain, NICD, which then is able to act as a transcription factor, migrating into the nucleus, and here interacting with nuclear proteins to activate transcription. This is in principle all you need to know. So it's in principle rather simple, but as you can see, there are many, many different molecules that have been identified, and many of them are mutated also and causing also valve defects, at least in mice or in other model organisms. And they have been also implicated in disease also by now, in valve disease. This is just showing you the context of Notch again. So Notch plays a role controlling epithelial mesenchymal transition, both in the AV cushion as well as in the outflow tract. Notch in particularly controls the expression of transcription factors that control the expression of adhesion molecules. So SNAIL1 is a repressor of adhesion molecules, and it's controlled by Notch signaling, and thereby the cells are able to leave the epithelium, the endothelium, and migrate here into the cushion area. All right, so the next step, now we are here, we move from here to here, so we have now cushions, but we do not have yet really valve leaflets. So how we generate those valve leaflets? So certainly those cushions are only precursors. They still have to be extensively remodeled. Cushions have to fuse, and this is depicted here. So I mean, those cushions are generated. In the AV tract, there are two, an inferior and a superior AV cushion and two lateral ones. And a similar situation is here in the outflow tract, where you have two outflow tract cushions, which are subsequently separated by some myocardium. I don't want to go into detail here how this is all achieve, but it's also a rather complex process. So those cushions are fusing, as I said, and so you get then a cushion complex, as depicted here in this section, and this is then subsequently further remodeled to generate out of that valve leaflets. So the central part here generates the blue-yellow valve leaflets, while the lateral ones shown here and shown here are generated by the lateral cushions. So there's remodeling of cushions to generate the leaflets. So what are the cells that make up the cushions? To answer this kind of question, where are those cells are coming from? Are they all endocardial cells? One uses nowadays fate mapping, and the modern research mostly is done in the mouse, where you can't really simply label individual cells. But what you do is a genetic labeling called Cree-mediated fate mapping, and it's shown here. So you have on the one hand, in a mouse, a tissue-specific promoter that drives an enzyme called Cree recombinase, and you have a reporter gene which contains two target sequences which are recognized by this enzyme. And if both genes, the construct, the reporter construct and the Cree construct are in the same animal, then recombination occurs, and you generate then an active reporter gene, like, for example, LAG-Z. And the LAG-Z can be then visualized by blue staining. As you see here, the heart becomes then blue when the two genes are together, because then the recombination occurs. In the absence of the Cree recombinase, only the reporter construct is there, but there's a stop signal here which needs to be removed that this becomes active. So this mouse embryo is therefore unstained, and also on sections you do not see any labeling. So important is now that we have here a tissue-specific promoter. So for example, in the myocardium, so only when the myocardial cells are formed, then the recombination occurs. In no other cell population, there will be recombination. So you genetically label cells when they are formed, and any offspring of these cells will also be labeled. So you can follow those cells during further development. So this kind of experiments have been done also in the case of the valve. So we have, for example, used here tied to Cree, which is an endocardial promoter, and promoter that is active in endocardial cells. And then we can see here that the entire AV cushions are labeled, and also later on the leaflets, the valve leaflets, are also nicely labeled. And this is very prominent in the AV valve, but less so in the outflow tract valves, which are only partly labeled, which indicates that not all cells that make up the outflow tract valves, the semilunar valves, are derived by the endocardium. So to clarify this further, we need to introduce you to this picture. So we have on the one hand the heart fields that are indicated in the beginning that generates all the muscle cells and also the endocardial cells. So muscle and endocardial cells are generated by the heart fields positioned here. But this is not the only cells that make up the mature heart. There are at least two additional cell populations. One cell population is the cardiac neural crest that migrate via the outflow tract, and particularly is important for separating the aorta and the pulmonary trunk. And we have the proepicardium, which generates a quite variety of different cell types, including smooth muscle cells, endothelial cells, fibroblasts, epicardial cells, and even intracardiac blood cells. And these are also migrating on the myocardium and generating important structures and cell types of the myocardium. So I'm saying that to you because in both cell populations, the neural crest cells and the proepicardial cells contribute to the valves. So this is shown here. Shown here is an experiment used again with another Cree, which is now a Cree that is active in the proepicardium, shown here in red. And then if you follow this over time, you can see that there are leaflets in the AV valve that are populated by those epicardial cells. So in particular, those lateral AV valves are populated by epicardial cells. Is also shown here. So here in red is nicely the labeling of this lateral parietal leaflet. In both cases it's labeled, while the other one is unlabeled. And the other thing is the neural crest cells. The neural crest cells in particularly play an important role in populating the outflow tract cushions. This is depicted on the next one. Oh no, there's first shown again the migration pathways. So the neural crest cells are generated here in the neural tube, and they're migrating through the aortic arch arteries into the outflow tract area, and they are involved here in separating aorta and pulmonary trunk, but they are also migrating further and populating a part of the valve apparatus in the outflow tract. So you see those blue cells here are all fate mapped and are all originating from the neural crest lineage. Okay, the final part of my talk will deal then now with the role of blood flow, because I mean, we have so far discussed signals and genetics, but we have also clearly another regulator that is involved in valve development, and this is mechanical in nature. So I mean, endothelium is known to be very sensitive towards changes in shear stress. And the first indication that this may play a role in valve development is an experiment which has been done by the Leiden group, by Adriana Gittenberger de Groot's group in Leiden. And what they did was simply modifying the venous return, so the amount of blood that is returned to the myocardium. If you change it by placing a little clip here in the chick embryo, you will affect the blood streams that are entering the myocardium. And shown here is what those embryos will then generate valve anomalies, which both affect the valvular apparatus quite significantly with a high number. And to show you a little bit more about that, I'm just showing you here a video from a zebrafish heart, embryonic heart. What you see here is it's a rather simple heart, which starts in here's the atrium, here's the ventricle, and here's the outflow tract, the bulbus. You can see here that it's clearly the blood generates, is most likely be very informative for the endothelium, and is certainly a morphogenetic factor for generating valve structures. So this is simply the biomechanical shear stress that is visualized here. Now it's filled up the ventricle, and now we see the vectorial transport through the outflow tract. So this has been now further analyzed in the zebrafish in a nice way, and I hope this, yeah. So the first experiment was very simple, and that was simply by placing a little bead and blocking the filling, or either the sinus was blocked or the outflow tract was blocked. And this is just a placement of a little bead here as a control. So normally both an AV valve and an outflow tract valve is formed in the controlled implanted embryos, but if you block the venous inflow, as well as when you prevent the blood from leaving the heart properly, you mess up valve formation. So they don't have a proper valve formed here in this case. So there's certainly an effect on the valve apparatus by this rather crude method. This is just some movies showing you an important point, and that is in an early embryonic heart. So in green here is the endocardium shown. In an early embryonic heart, there's a significant amount of blood that is actually pumped back into the atrium. So not all the blood is transported in the proper direction. And this is less and less the more mature the zebrafish heart becomes. So there's only a little bit of blood that leaves retrogradely the ventricle back to the atrium, and there's no blood pumped back in the mature 5-day-old fish heart. And there was a realization that maybe this backflow, this reverse flow, maybe is has an informative value and maybe regulates valve growth. So shown here is the amount of backflow at different time points. You can clearly nicely see how much blood will actually leave the ventricle again and goes back to the atrium before it's then pumped out. And so initially, during the first stages of development, there's a significant amount of backflow, which ultimately, as I said before, at 5 days, it's completely gone. And an experiment to prove that was then to change a little bit the blood viscosity. So what they did was to play around with certain genetic methods to prevent blood formation, and thereby changing the blood viscosity, which then certainly has an impact on the biomechanical properties of and the effect on the valve endothelium. And when you mess up with the backflow, then you get a loss of valve tissue. So normally you have those valve leaflets here formed after 120 days, and you can see here that with reducing the backflow, you clearly have a less well-formed AV valve that is formed. So clearly the amount of backflow is a determinant for the formation of proper valve leaflets, which also makes sense, because I mean, if the embryo is able to sense that this is not yet perfectly sealed, then all what it needs to do is to work towards sealing bring this to sealing, and then you know you have done your job. And so it makes perfectly sense that the system works like that. The sensor for that is at least one gene has been identified, KLF2A, which is a flow sensor also in the endothelial cells, and that is clearly expressed at the site of the valve endothelium, both in the aortic valve and the mitral valve, just at the position where it should be to be acting as a flow sensor. When you inactivate the gene, then you mess up with valve development, and clearly there has been now a pathway initially worked out how this may be working. I don't have the time to get into detail. There's clearly different mechanisms that may be involved in the actual physical flow detection. A prominent one are cilia that are present on endothelial cells and that lead then to calcium influx in the cell, but there could also be stretch-sensitive ion channels and multiple other membrane-localized sensors that may be involved in this process, and there's still room for quite a number of research with regard to mechano-signal transduction. We are now at the stage that we have now valves, but we still need to improve them in their morphology, so there still has to be further growth generated, and those pathways involved are, again, VEGF, Wnt, TGF-betas, BMPs, FGFs, different growth factors that have been identified, but remodeling and further growth is far less understood at the moment, mainly because we do not have very good assays, so we don't have a very good system to work out what those different factors might do in this process. There is certainly also postnatally further remodeling going on. This is just shown here that in fetal and postnatal valves, there is a reconstruction of the extracellular matrix, so collagen accumulates in specific parts of the valve leaflets, and certain genes are modulated during further developmental stages. So clearly, there is a lot of processes going on also postnatally. And just as an introduction to the now more disease-oriented talks, clearly now the valve can change their fate due to multiple reasons. So there will be then an activation of a pathway that involves then further a different program, particularly the accumulation now of calcification in the valve. And that involves in particular cell type, which are the valve interstitial cells, which have been identified to be particularly sensitive to changes in flow and are the culprits that causes the calcification. And an important and not yet clarified question is where those valve interstitial cells are actually coming from. Are they endocardial, epicardial, neural crest derived, hematopoietic stem cells derived? There's certainly clearly room for further analysis where those cells that are involved in valve, pathological valve remodeling, where they're actually coming from. And of course, there's also a disease where we will hear a lot about, bicuspid aortic valve disease. And interestingly enough, several of the genes that I mentioned, in particularly the gene Notch1 that I've indicated to you that plays an important role during epithelial mesenchymal transition, has been also identified to play a role in bicuspid aortic valve formation. So clearly, identifying the pathways and understanding how valve development occurs will also help us to understand valve disease at later stages in development and postnatally. I think that's it. Well, we'll leave the summary for it for the time being, and I thank you for your attention.
You had your heart tube with almost like a peristalsis movement, sending blood maybe.
Yes.
At what stage does it actually start to effectively beat? And do you have?
Oh, at that stage it beats.
That's beating.
It's already beating by that stage. Yeah, yes. It simply uses the differences between the vessels, deformation ability versus the heart, which obviously because of the cushions of the cardiac jelly, excuse me, it's more changeable, and so it's not so rigid. So I mean, the contraction obviously causes a wave that is sent in both directions, and because of reflection, because it reaches the venous vessels earlier than the aortic vessels, you actually get a directional flow. So it's a trick to generate the directional flow without valves. And that is obviously working fine as long as the heart is very simple and the embryo is small. If you're getting bigger, you need a better apparatus, and that's when the cushions are coming into place, which are now a little bit better. And if you remember the zebrafish, where you see these progressions, so you need a more and more perfect valvular apparatus, which starts from a very simple jelly towards a cushion, which then goes in size. And then you need leaflets, which are still leaky, and then this will be further and further improved, and thereby your embryo can grow further and further in size. There's certainly a limitation. The moment your valvular apparatus doesn't perfect, you will die.
Question?
What's the impact of this fascinating work on the clinical practice? And knowing that there is a gene for the bicuspid valve, and because of calcification, do you mean that in the future we're going to change the gene and have it as a clean bicuspid, or will it go back to being bicuspid?
Well, of course, I mean, it starts with understanding disease, and the next step may be developing towards therapies. But I mean, if you think, for example, nowadays we have small molecules, we can screen, let's say, for example, in the zebrafish, screen genetic chemical libraries to modify, let's say, a defect in the valve by simply placing those chemicals into the aquarium, and then screening for one which will improve the process. And that is towards actually functional therapy. I mean, I'm not so sure about any genetic therapy, which so far has not been really very successful. But I would think that those chemical screens may be way forward. I don't know whether this will succeed. I mean, certainly once we have identified the players, we can simply work towards therapy, and it's a matter of working hard on it, and we will probably succeed at some stage. But the main thing is to understand first the process, because I mean, we do not know what causes bicuspid. Is it too little, less amount of cells? Is it the matrix? I mean, we have identified Notch, but we do not know what is actually Notch doing in this process. So we have to understand why this mutation is causing this phenotype.
Do you think in the future it can present?
I don't know. Hopefully, yeah. I mean, who knows? I mean, it's a matter of if we understand it, will we have a way to change it? As long as we don't understand it, there will be only imperfect ways to treat it. So a way towards causal therapy is understanding, I think.
Can I ask a quick question? As a clinician, one thing that strikes cardiologists is that the aortic valve and the mitral valve look a lot different. in the structure and in function.
Yeah.
So is that mostly acquired because of different mechanical stress, or is that also due to developmental reasons?
Well, I mean, they certainly are formed differently from the beginning. I mean, if you see the cushions have a completely different morphology. There's certainly, I haven't alluded to that, but clearly also the biomechanics are clearly different in the outflow tract versus the AV valve. And of course the cells that immigrate are different. They are made from different myocardium. So from the beginning they are different. And all that may matter, to what extent, of course, is a matter of finding out.